Hello and welcome everyone. Thank you so much for watching our talk here today, presented at PEGS Boston Virtual, hopefully the last one. My name is Anthony, and I'm the Director of International Business Development here at Rapid Novor. Today, we'll be taking a journey through the world of antibody protein sequencing. As a quick agenda, we're going to go over briefly who we are. We're going to look at some antibody discovery workflows. We're going to talk a little bit about the REpAb workflow, and then we're going to examine some case studies and future directions for our team. So who are we? Well, Rapid Novor has been in the business of proteomics software development for nearly 20 years, and it was just over 6 years ago that they decided to focus specifically on the issue of sequencing proteins by using mass spectrometry. In the past 6 years, we've been able to help over 400 different clients worldwide, many of the major pharmaceutical companies by revenue, as well as major academic institutions such as MIT, Harvard, UCSF, Oxford, and Cambridge, as well as many more all around the world. And in that time, we've sequenced over 3,000 monoclonal antibody and proteins. Of course, most of that work was completed through our REmAb, monoclonal antibody and protein sequencing platform. But today we'll be talking about REpAb, the polyclonal antibody sequencing platform that we've been working hard on in the background. And in the future, we hope to talk a little bit more about other services, in particular NovoRig, which is useful for providing a relative quantitation of proteotypic peptides of interest, which might be useful for biomarker work or vaccine studies and the like. In addition to CheckMab, which is useful for confirming sequence similarity between a batch A of an antibody and a batch B of an antibody. But we don't want to talk too much about that. We want to talk a little bit more about what we're doing at our core. So Rapid Novor fundamentally is turning the central dogma of molecular biology on its head. To quickly summarize what I'm talking about, we're talking about going from DNA, transcribing it into RNA, and then translating it into a protein of interest. Of course, the dogma dictates that this is the way it always happens in nature, and it only happens in this direction. But we really feel that we're also a part of nature. And in fact, we start with the protein of interest and generate a primary amino acid sequence, which allows us to then go back and create DNA and then go through this whole process the way that it usually does in nature. But this is fundamentally what we're up to. And it's enabled us to evolve antibody discovery. In the past several years, we've been able to sequence many different monoclonal antibodies from many different species of animals, including mice, hamsters, sheep, bovine, rabbits, canines, and llamas, amongst a few others as well. And we really feel that by applying our latest technologies in REpAb, we're really evolving the antibody discovery space. So away from just your mouse systems and hybridoma technologies, and really expanding that further and taking advantage of the unique immunology that some of the different species in this world have to offer us. In particular, today we'll be talking about rabbits, but this is really how we view it, is that we're agnostic towards the species of interest. Fundamentally, we're sequencing peptides and putting them back together into a full-length protein. And that's how we really feel that we're able to successfully evolve this antibody discovery space. But of course, to really comment on how we're changing things, it's useful to remember what we're already doing. So I'm sure many of you are more familiar than I am with hybridoma technology. This is the mainstay of the industry. It's been around since the 70s, thanks to Kohler and Milstein, who successfully were able to take B cells, fuse them with myeloma cells, and generate hybridomas, which spit out powerful antibodies of interest. And of course, many people have been able to leverage display libraries as well. In particular, phage displays become very popular over the past couple of decades and generating many successful antibody projects. Yeast display continues to evolve and generate many powerful new options as well. Both of these technologies do have drawbacks. In particular, hybridomas, rely on the hybridoma itself. And should it ever change or permutate or run into issues in terms of its genetic makeup, you might have a different antibody on your hands on a later point as compared to the original time point that you started. And phage display, of course, generates powerful antibodies, but they often require a lot of resources in order to generate and then subsequently optimize against your project of interest and your context of interest. And of course, we have B-cell cloning, which has been successfully working to deliver powerful antibodies directly from patients of interest and animals as well. But in a lot of cases, in particular people that we speak with, we find that they're left with a vast ocean of data in the absence of any kind of antigen cell sorting. And in the case where they are able to successfully sort cells by antigen positivity, you might have a lot of attrition of those leads as a result of those efforts, and in an attempt to successfully clone them and then re-engineer them. So all of these methods do work well in their particular context of interest, but of course, naturally, have drawbacks. And we really feel that we are in the position to offer some complementary options to some of those drawbacks. In particular, let's say you have a hybridoma of interest and you're trying to clone it, but you've run into an issue with respect to the genetic data. Maybe you're actually cloning the wrong chains. Perhaps there's actually a artifact in terms of an additional light chain or additional heavy chain present in that hybridoma cell line, and you're actually unable to pull out the correct chain of interest in order to successfully recapitulate the binding activity of that antibody. In fact, you could start with the protein and leverage a workflow like we have here in REmAb, wherein we start with the protein of interest, and after a quick quality check, we're actually able to digest that full-length protein using a multi-enzyme cocktail, and then generate many different peptides, which are able to then be put through our LC-MS platform and generate spectra, allowing us to then interpret the primary amino acid sequence of those peptides. Once we have enough peptides that can cover the span of the entire protein of interest, we're then able to assemble all of those peptides back together into your full-length IgG or protein molecule. And it's from this basis that we really want to extend the technology further and into more complex situations, like in particular polyclonal antibodies. So without further ado, let's talk about those polyclonal antibodies. Well, naturally, it's fair to start off with a starting point, which is the workflow that most people find themselves using. So in a polyclonal, really, we want to highlight that people often use them for a couple of key reasons. I think the main reason is honestly the high levels of affinity that you can get out of a polyclonal antibody, which fundamentally come from the broad biophysical diversity that is generated within that poly antibody. You don't just have one antibody binding against one epitope on one antigen of interest. You have several different or dozens of different monoclonal antibodies that are binding against multiple different points on that antigen of interest. And that is what effectively enables a higher level of affinity as compared to some of the monoclonal antibody options that you might already be utilizing. And of course, with conventional monoclonal antibody generation technologies, they can be quite resource intensive. As opposed to starting with an antigen, taking a bleed, and purifying out your PABs, you have to go through this process of extracting the B cells, fusing with the myeloma, doing your clone selection, and then purifying out your monoclonal antibody of interest. That leaves you with a supernatant or potentially a purified antibody of interest against your target, and hopefully that works really well from you. However, having an antibody at one time point isn't enough to ensure the long-term success of your project. And in fact, many people have decided to go with, naturally, gene sequencing in order to immortalize their antibody of interest. When you can get the gene sequence, recapitulate the antibody through its genetic material, you can create a recombinant and then indefinitely have access to that antibody of interest. But in the absence of that genetic material, as I mentioned earlier, in the case of a hybridoma, which maybe has been sitting in a freezer for 20 years, or in the case where your monoclonal antibody actually isn't, and there's another cell inside of that well, you might be cloning the wrong chains, and it might be actually quite difficult to recapitulate that original antibody. You can start with the protein, and this is how REmAb and Rapid Novor has been able to offer complementary technologies in these antibody discovery workflows. But it doesn't stop there. We've acknowledged that PABs really have a powerful role to play in the industry, and they have for many years. And that doesn't stop today. We really hope to actually complement these existing technologies by doing something similar, wherein we start with your antigen of interest, put it into a rabbit or your host organism of interest, extract the bleed, purify the polys, and then go through this process of sequencing the most dominant antibodies that we can find in that polyclonal mixture, allowing you to recapitulate the original binding activity of that PAB by expressing a cocktail of mAbs or just one mAb in particular that happens to be a rock star. So naturally, with PABs, you find that they're easy to produce. There's already a lot of them on the market that can address tough targets. They inherently have a high affinity and broad biophysical diversity, which allows them to be very robust in many different applications. But naturally, they have drawbacks as well. There's batch-to-batch variation, there's a limited supply, it's finite, you have challenging upscale production for the same reason, and their binding is not strictly defined and difficult to characterize, which can lead to issues with respect to regulatory bodies and wanting to be sure about how this antibody and antigen are interacting, right? But mAbs also have a powerful role to play, naturally. They're the kind of rock star of the industry these days. You have recombinant production that's enabled. You have assurances because you have a standardized process behind this production, and they're easier to characterize because you have one mAb binding against one epitope of one antigen. Hopefully. But of course they have some drawbacks as well. Initially, they have a higher upfront cost, and they can have challenges against some targets depending on the modality by which you're generating those monoclonal antibodies. And of course, this is how we intended to leverage our technology, was by combining the serum of the animal of interest, which is naturally generating antibodies that are bound against the antigen of interest, and then put that through our sequencing workflow at the tail end, generating heavy chains and light chains, which are naturally highly abundant in the sample set of interest and also antigen specific. Once we have actually sequenced those through the proteomics, we can also perform NGS data analysis on the PBMCs by doing a bulk RNA-seq, at the tail end of which we will have, again, heavy chains and light chains that are pulled out of that entire PBMC sample prep. And then we can marry the proteomics and the genomics together in order to generate antigen-positive antibody sequences. Once we have vetted the quality of that database, we can then perform proprietary workflow for pairing the cognate heavy and light chains into their full-length antibody sequence. And as a result, we're delivering full-length antibody sequences to our clients and users of the technology. As a quick project overview, we start like most companies that are doing antibody discovery by immunizing. And as I mentioned earlier, we often leverage rabbits, but I'll get a little bit more into why that is the case in a few moments. So like most immunizations, we do a naive bleed, we do a test bleed mid-schedule, we do a final bleed. We like to do an protein AG purification just to extract the IgGs. Once we have an IgG pool, we then subsequently perform antigen-specific affinity purification and enrich for just those antibodies. After we've immunized, we perform a screening step. And in the screening step, we're able to assess 5 different samples of interest. That could be 5 different rabbits in a cohort, or that could be a couple of different rabbits with multiple different purification strategies deployed against their serum. At this point, we'll be able to perform our de novo protein sequencing to assess these samples and see if 1 or 5 of which are going to be feasible for subsequent deep sequencing efforts. And in deep sequencing, it's naturally more of the same, more sequencing, leveraging our proprietary techniques for enriching for high quality data reads from the proteomics, as well as performing your bulk RNA-seq and NGS data analysis. At the tail end of which, like I said earlier, we're able to deliver full-length IgG sequences, which are paired with their cognate heavy and light chains, which are going to be naturally antigen specific and highly abundant in the serum of your host animal. After that, we have the capability to perform synthesis with some partners, and it's a straightforward synthesis project. We're doing gene synth, transient transfection, expression of those IgGs, and then shipment to the end user for clone selection. But it's always nice to remember that if you're kind of able to do the first phase, the immunization, or the synthesis, that's fine. We don't need to do those kinds of workflows. We can always work with you with just the sequencing protocols themselves. Naturally, I'm sure you're curious, how did we do? Well, like we said earlier, we try to be diligent about screening for the appropriate species. Mice are challenging to work with for us because they're so small. They generate very small, relatively small amounts of antibodies within their bodies. And so we opted to go with rabbits, but that wasn't the only reason. Naturally, we can do NGS sequencing on rabbit B-cell repertoires, which is great, but it's also important to note that rabbits are really popular choice for antibody development generally. They offer very high levels of sensitivity and specificity as compared to your average mouse mAbs, particularly in immunohistochemistry. They also can generate mAbs against human epitopes, which are non-immunogenic in rodents, and against small antigens, particularly less than 20 mer peptides, PTM variants of a peptide, as well as haptens, which is something that's unique to the rabbits as opposed to your typical rodentia family. They also have a unique ontogeny, which offer complementary antibodies as compared to a mouse antibody set. So even if you've already run a mouse antibody project and it's generated some good leads, but you would like to get some more and offer some complementary data, it might make good sense to switch between two species and offer a different set of antibodies against your target of interest. And lastly, they have excellent stability, in particular because they actually have an additional cysteine bridge between a couple of the intrachains. And so we've decided to take this information and deploy it against, well, the most obvious candidate in terms of use cases that we saw to us. Of course, there's therapeutic antibody development, characterization of the immune response, as I mentioned, is of interest to many people, and diagnostic development. But the first one we wanted to do was just take a PAB and see if we can make it into a mAb or a mAb cocktail, right? And we did that by taking a peptide and immunizing some rabbits with that peptide of interest. The data looked good. For those of you that have never seen proteomics data before, this is really strong data. We have achieved levels over 20x. And by that I mean we have over 20 individual peptides overlapping every single amino acid throughout this entire sequence, the heavy chain on the left and the light chain on the right. In particular, you'll note CDRH3, which I'm highlighting here. It looks great. We have great coverage all the way down through the antibody. And similarly, this other antibody we found, this is are called R3. We have excellent levels of coverage all the way throughout, and it meets our threshold for a successful project, by which we mean we felt good expressing it. And so we did. And these are the results. PD030, as you see labeled in blue, was outperformed by R1 and R3, and we were able to successfully deliver these antibodies as reagent tools internal to our team, and we use them quite regularly, and they work quite well. Naturally, we then decided to extend the technology further and try to work with other partners. And so we did. We had a customer come to us and work in a diagnostic application. They wanted to take a biofluid of interest and purify out a specific peptide. They wanted to enrich for this peptide so they could subsequently perform a quantitative experiment to determine the level of that peptide in their biofluid of interest. And this project was very time sensitive for them as well. So while we do typically do the proteomics workflow and then the NGS on the back end, there were issues at that time in terms of delivering the NGS from our partners. And so we actually only had the opportunity to work with proteomics, and luckily it went really well. As you can tell, the customer was very happy. They found that we delivered highly specific, high affinity antibodies against this target, and they were powerful and fit for purpose. And what do we mean by that? Well, here's some more data. This was actually some of the data that we achieved from only using proteomics. And I should point it out here and say that this is the first time that any team has ever been able to successfully sequence multiple monoclonal antibodies directly from a polyclonal antibody serum in the absence of any genetic data. We pulled out these 6 antibodies, and as you can see in red, you have the original poly mix. So the other antibodies that we pulled out were very good. Eventually, we did get our hands on NGS data as well, and we supplemented these 6 antibodies with another 6 unique antibodies, which also performed similarly to the graph I'm showing you right now. So let it be shown that effectively, Rapid Novor has been able to recapitulate the binding activity of this PAB by using only proteomics. And of course, we are continuing our efforts and moving into more complicated and useful areas, in particular therapeutic antibody discovery. This is a typical antibody discovery workflow. I'm not suggesting that we're necessarily ready for game time just yet, but I am suggesting that certainly antibodies that are fit for purpose in an assay of interest can be pulled from our REpAb platform. PK/PD studies, can sometimes get tricky and context specific in terms of the host species and demonstrating surrogate endpoints. And in fact, REpAb has been demonstrated to be efficacious in pulling out unique binders and powerful binders against those tricky targets. Of course, though, there is the argument that one could skip some of the necessary sequence optimization if one started with the end result that's coming directly from a human sample set, or a set of antibodies that are coming from a host of interest. In particular, like if you're working with a unique animal model because you're in vet health, it might be tricky to deliver, for example, canine antibodies that are fit for purpose in your assay of interest. And instead of using a mouse antibody in that assay, which could lead to off-target binding against that mouse antibody from the host that you're testing it in, you could use antibodies that are directly pulled from that animal and reduce some of that off-target antibody response. And of course, in by complementing antibody validation strategies, it can be hard to find a good monoclonal antibody. Let's say you've done it, you might actually want to immortalize an antibody by using REpAb and allowing yourself to ensure access to that antibody. But in the absence of even finding, a good monoclonal antibody, you might have to undergo an antibody discovery campaign. And naturally, we feel that we're able to offer some value in that area as well. So what does it take to get started with us? Well, really, we just need the purified polyclonal antibodies and some blood, or just the PBMCs. In the absence of those two materials, we find that it's nice to just have a conversation about it. If you just got the PAB, if you just got the purified material, let us know. We can always have a conversation about it. And if you're working with a unique species like goat, sheep, llama, whatever, again, just have a conversation about it, and we might be able to help you out. That is ultimately the goal of our technology. Of course, in future developments, we're going to do some more custom NGS work. We've got a custom NGS lab set up in-house now, and we are continuing to grow that facility as we continue to grow the company. We have now been able to improve our protein-only sequencing capabilities, and we aim to continue to improve those as we expand the use of the technology. We're delivering more antigen-specific leads and considering novel methods for immunization. So, in summary, Rapid Novor has been able to deliver a library of antibody protein sequences through the use of our proteogenomics and proteomics-only platforms, which have proven useful in early-stage R&D for therapeutics, diagnostics, and reagent antibody development. And we're seeking royalty-free contracts for the use of the technology in the pharmaceutical and biotechnology industries. And if you have any further questions, I'm always here. Again, my name is Anthony. I'm with the company for, just about since the beginning, and I'm happy to take your questions. And as you can note, this is a pre-pandemic haircut. These days, it's been grown out a little bit more, but I look forward to answering your questions about antibodies whenever you're